In this lecture, we're going to focus on how the muscle uses fat during exercise. These fatty acids can come either from the bloodstream, which have been derived from the adipose tissue triglyceride stores, or from the triglycerides that are stored within the muscle. The fatty acids from outside the muscle, first of all, have to cross the sarcolemma, or the plasma membrane of the muscle. They have to then diffuse through the cytosol, cross the mitochondrial membrane before they're oxidized within the mitochondria. The breakdown or the conversion of fatty acyl-CoA to acetyl-CoA is referred to as beta oxidation, and this occurs in the mitochondria. During prolonged exercise, we see changes in a number of aspects of fat metabolism. These studies have involved tracers, again, labelling fatty acids and glycerol to estimate the rates of lipolysis, which is the breakdown of triglyceride, the rates of fatty acid uptake, and the rates of fatty acid oxidation. And you can see in this slide, during quite prolonged exercise lasting about 4 hours, the progressive increase in the rates of lipolysis, and this will come from both adipose tissue and potentially also from muscle triglycerides, the increase in fatty acid uptake from the blood by the contracting muscle largely, and the increase in fatty acid oxidation. This is a somewhat complex slide, but it demonstrates the interactions between the oxidation of fats from the blood and fatty acids derived from the muscle. In a normal situation, over time, as I showed you in the previous slide, you can see a progressive increase in fat oxidation of fatty acids derived from the plasma. And that's associated with a breakdown of muscle triglycerides, which then declines as the increase in fatty acid oxidation from the plasma occurs. If you give these subjects a drug which blocks the mobilization of fatty acids from their adipose tissue, you don't see the same increase in fatty acid oxidation from these plasma sources. And under those circumstances, you can see an increased reliance on the intramuscular triglyceride stores. I should add, under this condition of low free fatty acid availability, as I mentioned in the previous lecture, that will influence the rate of glycogen breakdown. So not only is there an increase in muscle triglyceride utilization, there's also an increase in muscle glycogen utilization under these conditions to compensate for the reduced plasma fatty acid availability. The breakdown of triglycerides in both skeletal muscle and adipose tissue is referred to as lipolysis, and the enzymes that break down these triglycerides are referred to lipases. And there are two key lipases that have been identified both in adipose tissue and in skeletal muscle that are important in regulating the breakdown of triglycerides during exercise. The first is known as adipose tissue triglyceride lipase, and although it was initially identified in adipose tissue, it's also been found in skeletal muscle. The second important enzyme is a lipase known as hormone-sensitive lipase, and as the name implies, it's regulated by a number of key hormones. It's been shown that in the breakdown of triglycerides, that ATGL is primarily involved in breaking down triglycerides, and the hormone-sensitive lipase seems to attack the diglycerides. Both enzymes are activated in adipose tissue and in the muscle during exercise to break down the triglycerides, mobilize the fatty acids, and make them available for either transport in the blood, in the case of adipose tissue, or transport in the cytosol of the muscle across to the mitochondria. If we first look at adipose tissue lipolysis during exercise, there are a number of important regulatory factors. As I said, it's mediated by the lipases, ATGL, and hormone-sensitive lipase, or HSL. And an important activator of these lipases in adipose tissue is the sympathetic nervous system circulating adrenaline acting through beta receptors. One of the side effects of taking beta blockers, and a number of cardiovascular patients do that, is that they see an inhibition of the mobilization of fatty acids during exercise. And that can sometimes be associated with muscle fatigue, probably because of an increase in glycogen use, and that needs to be managed if you're employing exercise in cardiac patients who might also be on these beta blocker agents. The decrease in plasma insulin, which is important for the mobilization of liver glucose, is also important for the mobilization of fatty acids. And insulin is known to have potent anti-lipolytic effects, and so its reduction during exercise is important in allowing a normal increase in free fatty acid mobilization. The adipose tissue blood flow is an important parameter as well, because it continues to flush out, if you like, the fatty acids from the adipose tissue into the systemic circulation. And it's known at higher exercise intensities, where there tends to be some vasoconstriction of the adipose tissue, that can often limit the availability of free fatty acids to the contracting muscle. Because free fatty acids are hydrophobic, they have to be transported on albumin, the major plasma protein. And the ratio of the binding of free fatty acids to albumin is also an important determinant of the mobilization of free fatty acids from adipose tissue. The blood glucose concentration, both directly and also via effects on insulin, can influence the mobilization of fatty acids. And it's also known that lactate can inhibit fatty acid mobilization or lipolysis in adipose tissue. And perhaps at higher intensities, when you see an increase in lactate, that might also contribute to a reduction in the availability of fatty acids. And interestingly, one of the major metabolic effects of caffeine is to stimulate the mobilization of free fatty acids. So in addition to any effects that caffeine has on the central nervous system and acting as a stimulus, it's also known to increase free fatty acid levels, and that may contribute in part to the ergogenic benefits of caffeine in endurance type exercise. If we turn our attention to skeletal muscle lipolysis, then again those two enzymes are involved, ATGL and hormone-sensitive lipase. Again, the beta-adrenergic system and increasing in adrenaline acting through the protein kinase A pathway will activate the hormone-sensitive lipase. A calcium-dependent kinase known as extracellular regulated kinase, or ERK, is activated in response to calcium, which increases during muscle contraction, and that will stimulate lipolysis. The blood glucose concentration will tend to inhibit the hormone-sensitive lipase. And interestingly, the availability of free fatty acids, as I showed you in that earlier slide, if free fatty acid availability is reduced, then there'll be an increased reliance on intramuscular triglycerides. Now, for those plasma free fatty acids that need to be taken up by contracting muscle, for many years it was thought that this occurred by simple diffusion, and that by raising the plasma levels of free fatty acids, that would automatically increase fatty acid uptake into muscle. And to a large extent, that's true, but it's also been shown in recent years that an important part of free fatty acid uptake occurs by facilitated diffusion, in much the same way that we saw for glucose uptake. And so, the major determinants of skeletal muscle fatty acid uptake then are the plasma level or the arterial concentration of those free fatty acids and the ability of the muscle to take up and oxidize those fatty acids to maintain a diffusion gradient. There are a number of sites where fatty acid transporters are involved. A number of proteins have been identified. These include the fatty acid binding protein, FABP, another fatty acid binding protein called CD36, and fatty acid transport protein, or FATP. And these proteins are involved in transporting fatty acids across the sarcolemma, across the largely aqueous environment of the cytosol inside the muscle, and also across the mitochondrial membrane. In relation to that latter process, these fatty acid transporters, in particular CD36, do so in partnership with an important transport compound known as carnitine and an enzyme that's involved in that process known as CPT. And together, these transporters facilitate the mitochondrial entry of fatty acids so that they can be oxidized. As I mentioned earlier, the process of beta oxidation involves converting fatty acyl-CoAs to acetyl-CoA, and there's a key enzyme called HAD found in the mitochondria. And the amount of that you have will determine how much you can oxidize the fatty acids. I mentioned the compound carnitine, and it has an important role in facilitating the transport of fatty acids into the mitochondria. And it does indeed sit at the crossroads of carbohydrate and fat metabolism. And you can see an interaction between the two here. And if there's a very large increase in the rate of carbohydrate utilization and production of acetyl-CoA, then carnitine can act as a buffer, and you get increases in acetylcarnitine to buffer these increases in acetyl-CoA. One of the challenges then is that impacts on the availability of carnitine for fatty acid transport into mitochondria, and I'll come back to this point in just a moment. But you can see here, in relation to the mitochondrial uptake of fatty acids, here is the long-chain fatty acid, the importance of carnitine, and the CPT1 enzyme complex, which transports the long-chain fatty acid into the mitochondria, where it can undertake beta oxidation and enter the oxidative pathway. And so carnitine has an important role. And you may have noticed, if you've been to some of the sports nutrition shops, the promotion of carnitine supplementation to promote the burning of fat, both for weight loss and also in bodybuilding. And there's been some research over the years to suggest that perhaps carnitine supplementation can play a role. There are equally some results suggesting not a major role. But again, the important role of carnitine is to transport fatty acids into the mitochondria. And finally, how well a muscle can oxidize fatty acids is also a determinant of fatty acid uptake. And this will maintain the diffusion gradient into the mitochondria. And so here you can see the relationship between fatty acid oxidation, plasma fatty acid oxidation, and the concentration of that enzyme HAD, which is involved in beta oxidation. If you increase the amount of mitochondria in the muscle, you will get an increase in HAD. And as we saw in our muscle lectures, one of the muscle adaptations to endurance type exercise is an increase in mitochondria and an increase in HAD, and therefore a capacity to oxidize fatty acids. Why is it then that fatty acids and fat oxidation decrease at higher intensities? I showed you in one of the earlier graphs the increases in fat oxidation that occur at moderate intensity, but then a decrease in total fat oxidation as you go to higher intensities. You can see at the lowest intensity a heavy reliance on plasma fatty acids with a little contribution from muscle triglycerides. As you increase the exercise intensity, the contribution from plasma-free fatty acids becomes relatively less. There's initially an increase in intramuscular triglyceride use, but then a decrease at the higher intensity. Some of the factors that contribute to this, certainly in relation to plasma-free fatty acid oxidation, a reduction in the availability and the delivery of fatty acids can contribute. Inside the muscle, there are relationships, partly related to carnitine and CPT that I showed you, that as you increase the rate of glycogen breakdown, as you increase adrenaline and sympathetic nerve activation, that is known to inhibit the activity of CPT, and that will have a negative effect or inhibit the mitochondrial uptake of fatty acids. And for the reasons that I outlined, carnitine acting as a buffer of acetyl-CoA derived from carbohydrate as you increase the exercise intensity and increase the production of acetyl-CoA from carbohydrate, that can often reduce the availability of carnitine for fatty acid uptake. An interesting aspect of this, though, is that the oxidation of carbohydrate requires relatively less oxygen for a given amount of ATP production. So it makes good sense for the body to rely more on carbohydrate as you become closer to your maximal oxygen uptake. In terms of training effects on fatty acid oxidation and intramuscular triglyceride use, just as we saw a reduction in the reliance on carbohydrate metabolism after training, we see an increase in the reliance on fat. And so what we have here is the rate of fatty acid uptake in the open bars and the increase in fat oxidation. And you can see both in untrained and trained an increase in both fatty acid uptake and fatty acid or fat oxidation. You'll notice here that the increase in fat oxidation is somewhat higher than the increase in fatty acid uptake. And that reflects the increased reliance on intramuscular triglycerides, which you won't measure in this parameter here. So exercise training increases the oxidation of both plasma free fatty acid and intramuscular triglycerides and the fatty acids that come from them. In relation to the effects of training on muscle fatty acid uptake during exercise, we can see a relationship between free fatty acid uptake by the muscle and the arterial free fatty acid concentration. You can see that in the non-trained muscle, there was a saturation of this process. And that's consistent with the notion that there are these fatty acid transporters that are becoming fully saturated in terms of their ability to take up fatty acids. In contrast, when you look at the trained muscle, you can see the increase in free fatty acid uptake with an increase in free fatty acid concentration. And it's thought that this reflects the training-induced increases in these transport proteins within skeletal muscle, together with an increase in mitochondrial oxidative capacity and an increase in HAD and the beta oxidative capacity. This will drive fatty acid uptake into the contracting muscle. And the increased ability of fat to be oxidized within skeletal muscle is thought to contribute in part to why regular exercise helps in reducing body mass and particular body fat levels, because of this greater ability to oxidize fat in skeletal muscle rather than to store that fat in adipose tissue. So as we come to the end of this module on fuels, hopefully you've got an understanding of how carbohydrates, fats, creatine phosphate can be utilized under various exercise conditions. If we look at aerobic type exercise, this slide summarizes the interaction between carbohydrate and fat utilization and the influence of increasing exercise intensity. And if you have an understanding of the factors which influence the selection of carbohydrate and fat during this exercise, that's relevant for sports performance, but it's also relevant for exercise prescription to try and optimize the utilization of fat to manage body mass and body fat levels.